Hi everyone, this is my protein. It is called MCR1 and it is mostly located on the surface of cells, specifically melanocyte cells, and specifically on their plasma membrane. And it can enter deeper into the cell in certain scenarios. Mutations in humans usually result in red hair, but I was studying mutations in mice, which results in lighter fur in mice. I used some pom-poms and pipe cleaners and I guess Sharpie and paper and just like a board as my materials to depict my protein. This represents the primary and secondary structures. This is just a more zoomed in version, and this is representing the tertiary structure, which I will go into more detail of. MCR1 does not contain subunits like some other proteins. Instead, it is a single polypeptide, which I depicted through just one continuous strand that is all one color, as you can see. Also, these lines are a little bit thin, but there's one right here, one right here. Within this space is the intracellular section of the cell, and then outside is the extracellular areas. So my first model shows the primary and secondary structures. In my primary structure of MCR1, I used a lot of pom-poms, as you can see, to show just how many amino acids are in a single polypeptide of this protein. In this structure, I've also depicted the 7 transmembrane helices, which are the loops that you can see coming out of the intracellular section into the extracellular section. And then this primary structure and its sequences of amino acids is what dictates how this protein will fold and function. The secondary structure is composed of 7 alpha helical transmembrane domains that we can see spanning across the lipid bilayer of the cell. These domains are connected by the loops that I've depicted. Each alpha helix is capable of hydrogen bonding and also interacts with G proteins. And then I created a second diagram in order to depict MCR1's tertiary structure. It's just this little loop. And it's 3-dimensional, as you can see, and it shows how the protein curls and folds. In the tertiary structure, the helices fold so that the extracellular loops, which we can see in the first model that we just talked about, are able to bind to melanocortins, which is a protein in the eyes and skin that makes melanin, and the intracellular loops that we also are here. These interact with G proteins and signal for cyclic AMP production. MCR1 is usually what we call a monomeric protein, meaning it can function without forming quaternary structures, which is why I just have this tertiary structure. However, it can interact with G proteins in some cases and form quaternary structures, but since this is just focusing on MCR1, I didn't create a quaternary structure because I didn't think it'd be fitting for the depiction of this protein. And so here, this one, now we can move on to this. I created a smaller, more zoomed in depiction to demonstrate how the amino acid arginine becomes cysteine in this mutation that we're going to be talking about, which changes the peptide structure. So as you can see, this darker blue I used to symbolize how from one amino acid to the second, it remains the same. But then here you can see this is 2 different colors. It shows the change in the amino acid sequence, which might seem small in all of these amino acids, but it makes a very big difference. And also this little red pom-pom, I might come back to this, but this is the mutation, and this is just a zoomed in version of this tiny mutation. And then I'm going to talk about the effect that it has on this whole protein. So to depict the mutation that causes my stub lighter fur, it's affected and how it affects the structure of the protein, I added, like I said, this red pom-pom. It's supposed to be at position 67, or it's the 67th amino acid. I'm not sure if it's exactly at position 67. This was just the best representation I could give, but I just wanted to point out and make it known that it is the 67th amino acid that is going through this mutation. So this specific mutation that I'm going to be talking about is called R67C. And when the protein mutates, the amino acid arginine is replaced with cysteine, as I said, making it so that the protein can bind to the melanocyte-stimulating hormone. So it cannot bind to the melanocyte-stimulating hormone in order to activate adenylyl cyclase. It's a complicated word, but and as a result, cyclic AMP levels remain low, and the protein can't produce eumelanin, which makes the mouse's coat lighter. So if the mouse was making eumelanin, it would have more melanin and it would have a darker fur color, but because of this mutation, it can't make it as well, and so it has a lighter fur color. And this affects the function of the organism because it allows certain mice to only blend into specific terrains. So, for example, a mouse with this variant would not want to live in an area with a bunch of dark rocks because it would be easier for predators to spot its light fur hiding there. So with these depictions, some challenges and limitations that I faced, this single peptide has so many amino acids. I bought tons of little pom-poms, but it was hard to fit as many amino acids as there truly are in this peptide on this board. So this is just a depiction showing that there are so many amino acids, but there are a few more, and it does span a little bit farther in the actual peptide sequence. And then again, creating only one model was really difficult in depicting like the entirety of this protein. So that was a limitation I faced, and I decided to build 3 things to depict it to help explain best to you what this protein looks like and its functions. And then finally, when proteins fold, they fold very specifically to do specific things and complete specific jobs. And this protein is no exception, but I did just curl a pipe cleaner a few times. It's not very specific, but it is just to give you an idea of how this protein would fold. Again, it would be a lot more specific if it was the real protein, but I just had pipe cleaners. So those are my limitations. This is my protein. I hope this helped explain what this protein does and its mutation, and thank you so much for watching.